Sir,
Can
we
we
are
go
live
live
now.
now?
Yes, sir. You can start now,
So,
sir.
good morning, everyone. Good morning, students. It's also my privilege to deliver this talk to you for the aspect of research methodology part, or to introduce you to the field of pharmaceutical industry, to the field of the chemical industry, and also to my beloved students who are also from the life science background. So, I am Dr. Vaibhav Mehta. I am currently working as a consultant, having my own consultant firm named Jetagam Consultant. I am operating to provide consultancy services to the pharmaceutical industry as well as to the chemical industries. Today, what I'm going to talk to you is about what are the current trends which are actually required as a part of the skill when you pursue your future PhD studies or your higher studies after completing your masters. Now, one or two things which we need to be very clear is that these skills are absolutely necessary for the new generation of technological advancements when you are going for the job, when you are going to establish your individual industries, or even when you are going for a profession which is non-related to the scientific part. Okay. So I think that everyone can hear me properly. If not, then please just message that if my sound is okay and properly communicating. Also, if you can able to see the screen properly, please check it out. So
Yes,
let's
sir. It's
start.
properly visible and audio is very properly. No problem.
Okay, okay. Then we should start with the presentation. That the title is, as you can see, Research Trends in Skill Requirement for Pharmaceutical and Biochemical Industries. Now, in next 60 minutes or 65 minutes or even little bit of longer part, uh, I will be presenting you some of the case studies, some of the definitions to the field, and also some of the information which is relevant for your scientific career. Now, I am understanding that you are now enrolling for your higher studies. In particularly, you are enrolling for your PhD studies. So the students who are enrolling uh, need to have some basic understanding of the research criteria, the basic understanding of what those things are going on into the market, as well as the things which are necessary as per the new generation advancements. Okay. So I'm going to cover the topic which is related to pharmaceutical industry, where you can see that we will be discussing something about synthesis, isolation, characterization, as well as documentation. Now, this could be a generalized sentence or a word. It could be for the chemistry fellows as well. It will be for the life science fellow as well because they also need to synthesize. They also need to isolate some biological molecules. Even they need to characterize them. And eventually, into your entire scientific research career, you definitely need to do the documentation. Then we will slowly move on to the second part, which will be what are CROs, what are CDMOs, what are biotech industries. So this is something that I will give you a little bit of overview where the industries are. Now this is specifically for the students who want to go for the job for next 5 years or 10 years of their life before starting some new business or before going to some another field. So if you want to pursue few years of the job, then this is the industry for you. The third part is the patent and the regulatory aspect. Now, from the same word, you can understand that what this topic is going to cover. Then we will also move on to, in general, the chemical industries. So not the pharmaceutical industries, but how does the chemical industry perform? Now, chemical industry also includes the same thing. It includes basic chemicals. It includes biotechnology industry. It also includes agrochemical, perfumery, flavors, fragrance, and so on and so forth. Finally, we will also move to what are the next generation tech industries. So we all are coming from a science background, whether it's the bioscience or whether it's the chemistry or whether it's the physics or any part. Nowadays, there are new industries which are upcoming, which requires tremendous manpower who are highly knowledgeable into the field of inorganic chemistry of rare earth or battery technology and so on and so forth. Okay, so let's move on. This is the slide which I always present to most of the students, where you can get the help of the new generation data or the new generation softwares available to start your research career. Okay, previously it used to be that when you start your research academic career, you identify certain area of research based on your suitable supervisor or your research guide, then you do the literature search about that particular topic of interest. You gather the information over a period of 3 months or 6 months of time. And then slowly you move to the practical part or you move to the laboratory to pursue your individual research. Now, in this particular generation, since everything is available on ChatGPT or Google Gemini or XYZ softwares and everything is now online, it becomes little bit easier for the students to not go through the hard copies of the literature search, but rather you can do the literature search with the scientific part. So here in this particular site, you can see that there are a lot of softwares available, there are a lot of database available, where if you want to synthesize, so on the right side of the uh, screen over here, you see that these are the final products of the molecule. Now, some of the molecule could be biologically important, some of the molecule could be used in the perfumery industries XYZ. Now, if you want to synthesize this molecule, which raw material to use, what kind of software to use, whether the software will give you the entire literature search, whether that software will give you the synthetic methodologies available, all those things. So, this website, this particular softwares and this particular details exactly will cover all those information. Okay, look at this particular slide where you have something like what are the drugs which are discovered by the innovator and what are the generic drugs. Now, when we talk about the innovator company, so for example, Merck or Pfizer or Engine or Gillette, XYZ, these companies are called innovator companies. They do very advanced level of research to target a particular disease. They identify the cure of this particular disease in form of a molecule. This molecule can be a small molecule. This molecule can be a biological substance, or this molecule can also be a combination of several biological parts along with some um, small molecules. Okay. So when these companies discover, they are basically called as the innovator companies. This company synthesize this particular compound, they evaluate their biological data, and finally, after approval from the regulatory authorities, they can market this particular drug as a substance to cure the disease. Now, it could be a cancer drug, it could be a drug against diabetes, it could be a drug against hypertension, XYZ. Okay. Then, when we talk about the generic industry, what we assume is that whatever the innovator company has done in terms of launching the molecule, the same industry which synthesizes the same compound, but now they are either using a novel route to synthesize or they are synthesizing with a very similar route, but some steps are little bit of different than the original sequence. But the point is that the end molecule is always exactly similar structurally in terms of characterization as the innovator molecule. And to identify this particular compound as a generic compound, you need to prove that the biological data or the efficacy of this particular compound should be equivalent to the same data as the innovator. Okay, so this particular slide I have shown that, for example, this is a compound on the second part is called lesinurad. Then there is a compound on the lower side which is called palbociclib. So how to make this? And in the previous slide, I have shown that if I draw a structure like this, that I want to synthesize lesinurad. I give the command to ChatGPT or I give the command to the software. Now the software will evaluate, it will do the retrosynthesis, it will also identify the scientific literature which is available, and it will slowly guide you to identify a potential synthetic route. It will help you in identifying some scientific literature based on whatever is available in the software. So this is an example how you can start your research career. Okay. Similarly, here are the examples of another molecules as you can be seeing that some of them are the final products, some of them are the starting material. For example, this is a starting material for one of the pharmaceutical compound. This is also a starting material for another pharmaceutical compound, so on and so forth. So that software or that literature will directly give you that if you want to work on this particular area, this is the basic information which is available, and you need to go through this, you need to study them, and then finally you can plan that what additional research you want to pursue in that particular topic. Okay. Now, even this thing that in the pharma industries, basically the generic industries, what they particular do, they try to reduce the cost of the material compared to the innovator cost, because now you are synthesizing in a cheaper fashion. Some of the readily available starting materials are cheaper, and even the patent rights or the regulatory rights after the expiration of the patent, there will be a lot of material available in the market from where you can start your synthesis. Now, I'm coming to the second part. So it is my request to all the students that please start making an electronic lab notebook of your research from today. Even if you are attending any kind of presentation, if you are attending any kind of scientific talks, if you are attending any kind of a discussion within your department, always keep a lab notebook, always keep a journal which is in the electronic format, or if you want to write, you can just write down, but then again transfer it to the electronic copy. It's absolutely necessary in this generation to have a record of your ideas so that in future, whenever you have performed a novel research, whenever you have performed a research which is used for the publication part, whenever you have performed a research which is used for the patent part, the entire data will be collected based on this lab notebook only. So it is not necessary that you need to go for a scientific subscription or a paid subscription. You can have your own electronic lab notebook. For example, try to design a Word file where you have your experiment number, your date, your subject, your details, everything ready. So at the end of the day, whenever you have completed your experiment part into the laboratory, or whenever you are free inside the laboratory while your experiments are ongoing, you can start filling the details inside this Word file. Then you can upgrade this Word file from time to time, and then you can store the data based on the month, based on the year of your research, and based on even on the topic of the research which you are undertaking during your PhD. So the electronic lab notebook works as a accumulation of all the scientific and characterization data of your 3 years of your research. We in India don't have a very stringent condition to record all this data. But if you talk about US, if you talk about European laboratories, or even if you talk about very advanced laboratories in the world, most of the students and the professors are holding a electronic lab notebook because this record remains a permanent record of your research for the lifelong part. So even if there is a senior student who has performed some research before you can easily get that information from this kind of a lab notebook. You can try to understand the data, you can try to understand their synthetic and characterization parts like that. So please try to have your own electronic lab notebook. show that lab notebook, the format and all those things with your supervisor, with your guide, and have a habit of updating that particular document on a daily basis, or if not daily, on a weekly basis. That in a week, two times or three times you update your lab notebook. Now, just to showcase even the importance of that particular part, you can go through this below reference. Sorry. Consideration of implementing electronic lab notebooks in an academic research environment. Now, this particular article perfectly discusses why it is necessary in an academic environment to preserve your idea, to preserve your scientific thinking. Now, again, as I said, that if you have that particular record of scientific part in the form of a lab notebook, that will be eventually lead to a patent, it will lead to a publication, it will lead to any other particular intellectual property right for the rest of your life. And even if the publication, the record can be visible by rest of the world, so the world will know that whatever research you have done, whatever advancements you have performed in your career, that will be documented as a publication. Of course, we can go through the details about how to publish and all those details at the end of this particular presentation. Now let's come to the second part, which is CROs, CMOs, CDMOs, CRDMOs, and biotech industries. What are CROs? As it is mentioned over here, it's a contract research organization which functions by providing outsourced scientific, clinical, and business expertise to pharmaceutical industry, biotech industry, and medical device companies for drug development and research. Now, this is the general definition about an organization which performs a task based on the contract which you are providing to them. Now, this contract is a binding to perform a research. This is not contract for the manufacturing. So when we talk about CMOs, that's a contractual manufacturing organizations. So there in the contractual manufacturing organizations, you have contractual agreement, you have expertise in terms of the scientific part, you have the flexibility to utilize the resources. Then there will be a coordination between several different organizations who are working with this industry. So, for example, if you are into the academic career, you are collaborating with an industry and you are also collaborating with the pharmaceutical industries. Now that pharmaceutical industry is taking the idea from your academic research, they are developing that idea into a potentially drug molecule or a drug substance, and then they are outsourcing that particular research to an organization who can help them to make large number of molecules, to make lot of studies in terms of the clinical data, in terms of the characterization, identification, so on and so forth. Okay. Now, what are CDMOs? CDMOs are nothing but they are contractually development manufacturing organizations. So the organizations who are doing the research, who are doing the development part of the research, and who are also doing the manufacturing part of the research. Now, in that particular criteria, what they do the production, they do the quality compliance. So that means that whatever you are outsourcing to them, they have to comply to the regulatory bodies in terms of quality. They should also have divergence in process chemistry. So there should be a lot of different ways to manufacture that particular compound. There should be various different clients to pursue that part. And of course, we can also talk about the specialized equipments. So, for example, if your research requires any specific kind of a instrument or a very specific kind of a characterization technique, those all details will come into the criteria of CDMOs or CRDMOs. Now, the function of this particular organization is basically very basically, you can say that they are only used for reducing the cost of the synthesis. They help in deliver this particular compound to the market. So they work very fast. They work in a very planned manner, and they do it the science in a very designed fashion that the end product should be delivered based on the dates and the times. They also have something called a risk mitigation. So in case if you are working on a very, very large project where a lot of funding is involved, lot of monetary power is involved, there you also need to identify that in future what the risks are. So this industry will also help you in identify the risk mitigation part. And then they also have very specialized expertise in terms of doing the marketing research and in terms of launching your product into the market. This kind of organizations also help you in terms of target identification, then validation of the target, lead generation, clinical studies, phase trials, etc., etc. So now, if we identify, we have a pharmaceutical industry. Within that pharmaceutical industry, we have the innovator industry, and we also have the generic industry. Parallelly, we also have something called a contractual research organizations like CROs or CDMOs and the functions of this particular. So these are the two different industries which are operating into the domain of science, particularly chemistry and the life science part. So in general, whatever I am talking about over here, this applies to both the faculties as well as to the students who are involved into the life science as well as the chemistry part. Now we come to the patents and regulatory affairs. So this particular part includes like whatever your intellectual property are. The basic understanding of a PhD is that you are performing a research which is particularly unknown to the scientific world. So the first criteria that your research should be completely unknown. The research should be completely novel. So that means the research which you are performing, the results which you have generated, and based on the results, whatever conclusion you are forming at the end of your research topic, that conclusion has to be completely novel so that you can get a PhD degree or you can get a degree to pursue your future career where you have done something novel in the next 3 to 4 years of your scientific career. Now, when you have this patents and regulatory affairs, you also need to have approvals as per local guidelines of a drug regulatory office. You need to also get the authorization of sale and supply of this active pharmaceutical ingredient or biosimilars. You need to also have the kinetic studies to perform the bioefficacy. So this is all parts of a patent and the regulatory part. If you have something very potentially which is useful for the humankind, which is useful for the society as a drug substance or even as another substance, then this kind of data needs to be protected in terms of a patent. Also, there should be the physicochemical analysis. So, how the formulation of the particular drug is performed, how that particular drug is delivered to the body, what the physicochemical responses we are getting, all those particular details need to be there. Then there is market survey and utilization of the data, advancement in terms of incorporation of guidelines, so on and so forth. Okay. So, this is particularly related to the patents and the regulatory affairs. Now, since we do not have the means of the communication, uh, I think I wanted to ask a question that what idea comes to your mind when you see this particular slide? Is it a simple aromatic compound? Is it a compound with lot of heteroatoms? Is it a compound which has got lot of functional groups? So on and so forth. Now, let me tell you why I am talking about this particular aspect. So, the below portion, what you see over here, it's a very basic compound. It's para-hydroxyaniline or para-aminophenol. You can say whatever you want to say. The second part is isobutyl benzene, and the third part is your guanidine molecule over here. So, these three are very crucial component for our survival to get rid of the diseases. The first part is para-aminophenol, which is a basic raw material for synthesis of paracetamol. The second part is isobutyl benzene. This is a basic raw material for the synthesis of uh, ibuprofen. And this is guanidine part, which is the basic synthesis for metformin. Now in India, paracetamol, metformin, ibuprofen, these are used as a daily routine tablets within our household to cure our fever, to cure our pain, to cure the diabetes, XYZ, all those things. So after the COVID or during the COVID time, our Indian generic industries started to realize that we are synthesizing a lot of advanced molecules. We are doing a lot of innovation in terms of synthesizing these molecules in the cheaper way. But what we have forgot is that we have forgot to synthesize these basic raw materials, which is completely now imported through China and which now requires a lot of attention for our country so that we don't need to import this material, but we need to synthesize this material within our own country. So if any student who is interested after the PhD to not go for the job, but if they want to establish their own industry, they could look for this kind of a very simple and basic molecules. They should do some research in terms of synthesizing these molecules in the cheaper way. They can establish their own industries and they can help our advanced generic industries by supplying this kind of materials. Similarly, over here on the bottom part, you can see this is a molecule which is a final product for a agrochemical. This molecule is called a glyphosate molecule. It is used for cleaning any kind of extra grass on the field. So it works as a herbicidal molecule. The central molecule, of course, everyone knows it's a tryptophan. And the molecule on the completely right hand side, this is a molecule which is now a very powerful insecticide used into the agrochemical industries. So these are the advanced structures which are now available into the market. You could perform your research on this kind of molecules. Try to identify an area of research where after 2 or 3 years of your work, you should feel that your research should be directly implementable into the society. It should go to the manufacturing industry, or there should be some kind of a positive effect of the research. If you publish, the publication should be useful to the scientific community. If you have a patent, your patent should give you some money. It should give you some rights for the legal part. You can establish your own manufacturing and like this. Similarly, if you look at this particular chart, this chart talks about the general chemical industry part where one of the companies which was established in 1869 and till today they are performing and they are the market leaders in terms of the flavors and the fragrance industries. The name of this particular company is DSM Firmenich. Okay, so you can look at those particular companies who are not into drug molecules, who are not into the biological part, but now they are into the scientific part of the molecules which we are using into our daily life, into the cosmetics, into the flavors and fragrance, into our perfumery products, XYZ. So even this industry requires a lot of attention in terms of inventing new biological molecules. The synthesis not from the plant based like extraction, but you can synthesize the same molecule now into the laboratory and then you have the same activity as the original natural product. Okay. Similarly, here you can see the large number of molecules which are available as a perfumery industries, and we are using these molecules in our day-to-day life. Most of the students are using fog or any kind of a deodorant. Try to look into the ingredients of those deodorants and see that which molecules are very close to your research, which you can perform. Similarly, from this particular slide, you can make out that this is related to a nutraceutical industries. The nutraceutical industries, any kind of additional supplements in terms of vitamins, in terms of proteins, even this industry is now upcoming industries for our Indian market. And the students who are interested to go beyond traditional pharma industries can look their research into this particular industry as well. Now, This is the next generation technology. This is particularly interesting for the chemistry students and the students who are coming not only from the organic synthesis background, but also who are coming from the inorganic part as well as the polymer industries. We have been listening to a lot of renewable energy, recycling of the very precious metal, recycling plastic, recycling our batteries which contains lithium and other precious metals. So these are the industries which are going to be upcoming in next 3 to 5 years within India, where your chemical career can be highly important. So if you don't want to go to the pharmaceutical industries, you can train yourself, you can perform the research which is implementable to such kind of a recycling or this kind of an industries where you have daily use of different kind of chemicals. You have to deal with the extraction of those chemicals, you have to deal with the processing of those chemicals, and eventually the precious metals are again converted back to the advanced battery materials. This is another industries which is the solar industries. Normally, as a chemistry or as a life science students, we don't see the importance of ourselves into the solar industries, but that's not true. Even in combination with the chemistry, in combination with the concept of very basic physical chemistry, you can go into this particular field of generating next generation uh, idea of solar energy, solar panel manufacturing, as well as inventing new technologies for harnessing the solar energy into our renewable energy. This is again related to the plastic industries, how to perform those recycling of the plastic, the waste bottle plastic, converting those plastic back into the form of, say, for example, monomers. You are again converting back them to the polymers. These polymers are now converted back into the finished product like resins, like the threads, like the bottles, and XYZ like this. So the plastic industries can also be used for the recycling part for up to 5 times or 10 times until you preserve the characteristic of that particular chemical compound in a very straightforward manner. Now, finally, whatever we have been thinking about this particular part, everything comes over here. This next generation tech industries or even the pharmaceutical or chemical industries, what they will require? They will require the help of the entire artificial intelligence. What is artificial intelligence? It actually covers three different parts where you have reinforcements of learning, you have a supervised learning, and then you have a unsupervised learning. When you have this three component comes together, you have a very deep learning. Now, with the use of a machine, you utilize this particular deep learning, and that entire concept of deep learning is now finally called as an artificial intelligence. So we use this AI based AI terms every day in our life, but the idea of AI is very deep rooted. It is not very straightforward. You need to think about the part that it involves a lot of learning based on the available data. You design a model, you run a program, you simulate those studies, you keep on optimizing that particular model again and again and again so that your efficiency of delivering the result or the output based on the query is now going to highest level of the efficiency. Now, if somebody from the students who have already attended my lecture when I was there in the month of October when Pakkavi Narasimha Mehta University organized a 2 day conference at Porbandar, I have shown this particular slides. Now, the next few slides is just an idea, uh, just that how the future labs are, how this particular things work. So as you can see that within the similar few wood, you have an NMR machine, you have a mass machine, you have lot of reactors over here, you have a IR machine. So within the same laboratory, within the same size of a room, your entire industry is running over here. Similarly, I have given this particular example that this is uh, based on the idea of crystallizing a organic molecule in a crystal lattice. So everybody of you know that this year's Nobel Prize was given to covalent organic frameworks. So this is the very similar concept that when you have a covalent organic framework, that framework is a culmination of metal part as well as the organic part. It combines together to form a porous structure, and within that porous structure, you can perform different type of uh, studies to harness the effect of that particular molecular organic frameworks. So in this particular example, you can use those MOF for identification of a crystal structure of a molecule, which is very difficult to identify through the standard NMR or any other techniques. Similarly, here you can perform the research with the use of very basic and cheaper raw materials. And this is an example of how the flow chemistry works. So these are the syringe pump which are easily and readily available into the market. You can combine your reagent into the syringe, and based on the flow rate of this particular flow meters, you are performing your reaction within this particular tube and you are collecting your final product within this glass. This is again an example based on the photochemistry, harnessing the effect of the photochemistry into our day-to-day chemistry life. So there are a lot of applications. You can get an idea. Later part, I will also kind of share this presentation so that you can, in general, have a lot of understanding of this photochemistry field as well. Again, nowadays in the pharmaceutical industries, we have such kind of a machine. This is a continuous flow photochemistry machine, where along with the flow chemistry, you are also having the light chemistry, and you are performing your reactions within this particular uh, reactor, and you are getting your final products. Again, this is an example that how to perform the electrochemistry within the laboratory. It's very basic, simple chemistry. Only thing is that you need to have your mind open. You need to have the imagination of your research idea, and with the very help of a very basic, simple equipment, you can perform very advanced level of scientific research inside your laboratory environment. But you need to be very creative, you need to be very patient, and you need to be very thinking about how to perform your research, which is novel. Now, let me give you one example about a very innovative drug which has been recently launched into the market. This is targeted to some chemistry students, but the students basically can have an idea, even from the life science, that how the industry performs. So, as you can see in this particular molecules, there are a lot of complexities. First of all, you have this 1,2,4 triazole heterocycle over here. You have this pyridazine molecule over here. You have a compound where there is a incorporation of a deuterium. So the drug substance which has got an isotope, it has got an isotope of a hydrogen. It contains deuterium within its structure. Then how many functionalities are present over here? You have this amide bond, you have this particular amine bond over here. Then you have this biphenyl ring, which is connected to the primary aniline part or the nitrogen part over here. So the complexity of the molecule is tremendous. Then you have a benzene ring over here, which is highly substituted on 1, 2, and 3 position. So that means you need to have a starting material where you require a lot of functionality, and those functionality needs to be converted to the final product. Now let me show you that how they have performed the discovery route. Discovery means when these particular molecules are designed theoretically, they are studied through the computer softwares as a drug-like product. These particular structures are given to the synthetic chemist. The synthetic chemist then designs the route to synthesize this molecule. And finally, when these molecules are synthesized, they undergo the biological studies as per their desired effect. So here you can see that discovery route, you are starting with such kind of a ester, methyl ester. benzoate molecules where it is functionalized with a methoxy in the nitro part. You are performing amidation, so directly converting your ester into an amide bond over here. Your amide now reacts with a protected dimethyl acetal. So you are converting an amide to an imine part over here. And then with the use of a hydrogen molecule, you are making this 1,2,4 triazole. So this is an example that how that triazole molecule can be constructed from very basic functional group like an ester. This kind of creativity needs to be culminated into your research criteria. When you perform your innovation into the research, when you perform a novel research in your research career, don't think that you need to go for complexity. Think about transforming the very basic functional groups into very useful heterocycles, which are requirement for the pharmaceutical industry. Once you have this 1 to 4 triazole compound, you are now alkylating, and finally you are reducing this particular nitro group to the amino group. Once you have this amino group, you are finally combining these two particular compound number 7 and compound number 12 in presence of this particular catalyst. You are getting your final molecule, which is deucravacitinib. Okay. Now using, see, 40% yield, 40% to 50% yield, very low yield compounds. So when you are doing the discovery, the idea is to generate the molecule as soon as possible inside the laboratory and on a smaller scales. Like you can have a milligram generation, you can have a gram scale generation, or maximum you require is few hundred grams. But when you have to go for the plant, you need to have particularly process chemistry part. That particular process chemistry needs to have a large scale manufacturing. So here you can see that in the commercial part, this is the final step. So this is the reaction of a chloro with the corresponding cyclopropyl amide. You are performing a Buchwald-Hartwig amination in terms of a palladium catalyst and a suitable ligand, and on a large scale. So from this particular part over here, on a 53% yield, now you are coming to 90 to 95% yield with a purity of 99%. Similarly, after having this particular intermediate, you are purifying the reaction and you are getting very high amount of yield. So here, this is the zinc salt. Now that zinc salt is now converted to the corresponding amine part over here. Okay. So here the final product you are getting in up to 80% yield and almost 100% purity, which is absolute requirement for the compound. So this is the difference from a process chemistry route. This is the difference into the manufacturing route. Now, as we have seen the formation of the heterocyclic molecule, here you can see that now I am synthesizing the same 1,2,4 triazole from the corresponding cyano part by reacting with this reagent, and now I am getting this particular triazole. So I am not starting with a ester, converting that ester into an amide, converting an amide into an imine, and then cyclizing and utilizing this reagent. So this kind of hydrogen hydrate, this kind of ammonia, these are very difficult to handle reagents. But if you have this particular reagent, very easy, single step, you are getting your final product, and then finally you are repeating the same sequence, and then you are getting your intermediate product which you want to use for. So this is an example that how theoretically a molecule which is done in the laboratory, when it goes to the commercial scale, the entire route of synthesis, the entire chemistry is now changed, but the end product remains the same, the purity remains the same, and our industry needs to now identify a cost effective route to synthesize this molecule for the anticancerous activity. Now, there is a small part which I have incorporated. So in the next 10 or 15 minutes, I will comment for specifically students who are from the life science department. When you talk about the life science department, we definitely talk about the biosimilars. Biosimilars, the compounds which are not the small heterocyclic molecule or small molecules which are synthesized in the laboratories, but these are the molecules which are isolated from the cell by cultivating those cells within the factories, within the reactors, and then you isolate your critical compound or the active ingredient which acts as a drug substance. So everyone should definitely know brand name Humira. Humira is the brand name of a compound which is now marketed for anti-cancer activity. What is the active substance? Adalimumab. What is adalimumab? Adalimumab is a compound which is identified as a monoclonal antibody. So it's not a simple heterocyclic molecule, but it's an antibody which now injected into our body acts as a drug substance to cure a particular disease which is present. Similarly, there are a lot of different active substances over here which are absolutely derived from the live cells. These compounds are purified, they are properly characterized, and then they are formulated to form a drug formulation which is now subjected into the market. So here, denosumab, filgrastim, rituximab, XYZ. So there are a lot of such kind of a compound. Even if you look into the market, you might have come across enoxaparin, heparin molecules, which are large molecules or large macrocycles, which are useful for different kinds of therapeutic areas. Also, when we talk about the biological or biosimilar parts, we also talk about the immunologics. What are these novel biological? So these are monoclonal antibodies designed to target specific receptors. Then you have the fusion of proteins and bispecific antibodies, which can improve the selectivity of the binding and which can also improve the efficiency of the compound inside the body. Then we have cell and gene therapies providing curative potentials, and we also have engineered cytokinins, enzymes, and even nucleic acid based structures. So, structures which are based on RNA and DNA. This is the slide which summarizes the different kind of antibodies, monoclonal antibodies, bispecific antibodies, antibodies which have been very specifically identified with the structural determination and what their function and roles are. So these are now what we talked about small molecules for the chemical or the pharma industries. These are the molecules for the biological industries. These are the molecules for the biotech industries. These are the molecules for the life science industries who want to pursue their further research and to work into the area of immunologics. Try to go through what are the definitions of monoclonal antibodies, how those monoclonal antibodies are synthesized, how those monoclonal antibodies functions, what are their function types, how they perform their functions within our bodies, all those details, and then you can advance your knowledge by going deeper into the research area, and then you can also target this particular topic as your research topic into your PhDs. Now, as I said, monoclonal antibodies. The idea is that this conventional monoclonal antibodies, their molecular weights are on an average about 150 to more than 150 kilodaltons. Then you have this kind of HCABs molecules, uh, biomolecules, which are lesser than 100 kilodalton molecules. And then we have nanobodies. Nanobodies are very specifically similar to the monoclonal antibodies, but now they are into the micromolar sizes or the nanomolar sizes, hence their name as nanobodies. What their specific features are, they are very small in size, approximately of 15 to 20 kilodaltons. They are very highly affinient, very easy to engineer, so you don't need a complexity to synthesize like monoclonal antibodies. They are very easy to engineer and synthesize. Even they can have a very large penetration within the body. They have better solubilities, XYZ. So there you can see over here that therapeutic areas, oncology, neurodegenerative diseases, infectious diseases, inflammation, toxicity, so on and so forth. Okay. Then we have this part which is called, sorry, uh, lipid nanoparticles. Now we have all come across COVID area. We have all passed through the COVID phase for 2 years of our life, where we came across several new different kind of concepts into our health and society. For example, what are vaccines? What is a COVID? How does that particular COVID affects a human society? Based on the health implementation of that particular effect, how to manufacture these vaccines, how to have these vaccines safer for our society. Now, when we talk about tablets, when we talk about injection, the formulation of that particular components are comparatively very similar and straightforward in comparison when we do a formulation of a vaccine. A vaccine requires very large number of components which combine together, and in combination they work as a drug substance, they work as a formulation to cure our diseases. So when I talk about a vaccine, one of the component of the vaccine is lipid nanoparticles. There are several different kind of lipid nanoparticles, as you can see over here: liposomes, nanoemulsions, solid lipid nanoparticles, lipid kind of polymers. So these macromolecules, from the life science, from the biological perspective, they are useful for the drug delivery. These molecules are not compounds which are showing their own activity. They are not the biological targets, or they are not the compounds which have their own biological activity. But they help the active compound to deliver within the body. So they are carriers to deliver active substance to our body. Then, as I said, mRNA molecules, so whatever nucleic acid based molecules. So, for example, if we have identified an mRNA, and that particular mRNA as a biological compound works for a particular disease, then how to deliver this particular mRNA vaccine in the body? So, you need to first collect this pure mRNA, then you need to combine this particular mRNA with the lipid nanoparticles. In combination with those things, you are now doing a formulation. That formulation will now be tested for the stability and for their biological studies. So there is a lot of complex science in the biotech industries, and in the biological field is also happening now. And as I mentioned to you in this previous slide, this all compounds, basically, these are all now generic compounds. They are not innovator anymore. And lot of another innovator compounds are also coming into the market. These all compounds, the biological compounds, along with the vaccines, they are all very complex science which needs to be performed based on uh, the advancements, based on the requirement of the society, and based on curing new and new diseases which are now we are seeing into the society. So again, this particular right part. So on the right hand side of the slide, as you can see, this is the structure of antibody drug conjugate. So not only that you are having an antibody, but you are also having a protein complex over here. You combine these two particular part, and now this entire concept work as a antibody drug conjugate. So this is also a drug substance, but this is not an mRNA part. This is a substance which combines an antibody, which combines a small molecule. That particularly two part comes together, and that two part uh, is performing some kind of an activity within our body, and in that particular activity we see a certain kind of a biological response, and that biological response is finally is the curing of the disease. Okay, so in general, this is the entire idea about the research which is ongoing into today's world. I have give you an overall introduction. Each individual topic which we have seen on the slide, we can have an entire 1 hour of presentation. So even this 25, 30 slides, you can have 30 hours of lecture to individually study each and every concept. But the point is to give you an idea, to give you a flavor about what is going on into the research area, how you can utilize this information to have your research uh, field identified, and how you can pursue your PhD studies with this sort of information. So, with this particular part, I am ending my presentation, and I will be more than happy now to take the questions from the students. And even apart from the common presentation, if you have questions which are related to say, for example, in general about your PhD, I will be very happy to answer them. So please, if you have question, as I was told that I have to read those into the comments box, so kindly put your questions into the comments, and I will be more than happy to answer them. Thank you very much.
Yes, sir.
So please take the questions if there are questions.
Yes. Uh, if there are any question, please ask in comment box.
Even in general questions also you can ask. You don't need to be very specific questions or anything. If you need some basic information, please feel free to have those particular questions also ready.
Yes. જો કોઈને કોઈ જનરલ ક્વેશ્ચન હોય રિસર્ચ મેથોડોલોજીને રિગાર્ડિંગ કે ઇન્ડસ્ટ્રીને રિગાર્ડિંગ તો તમે પૂછી શકો છો કમેન્ટ બોક્સમાં કમેન્ટ કરો.
ઇવન ગુજરાતીમાં પણ ક્વેશ્ચન પૂછશો તો ચાલશે અને ગુજરાતીમાં જ તમને જવાબ આપીશ.
યસ સર.
ઇવન આ પ્રેઝન્ટેશન ઉપર તમને કઈ કમેન્ટ્સ હોય, તમને કઈ ફર્ધર આમાં ઇન્ફોર્મેશન માટે તમારે સ્ટડી કરવું હોય, તો તમે તમારા રિસર્ચ ગાઈડ છે કે તમારે તમારા કોઓર્ડિનેટર છે એમને તમે નોટ ડાઉન કરીને જાણ કરી શકો છો કે પર્ટિક્યુલર ટોપિક કે પર્ટિક્યુલર પ્રેઝન્ટેશનનો જે આઈડિયા છે એમાંથી અમને શું શું યુઝફુલ થઈ શકે, અથવા અમે આ બાબતને કેવી રીતના અમારા રિસર્ચ કેરિયર સાથે ભવિષ્યમાં જોડી શકીએ. નો ક્વેશ્ચન્સ?
ના સર, નો ક્વેશ્ચન.
સ્ટુડન્ટ્સને વધારે ઠંડી લાગી છે આ સિઝનમાં કે પછી બધા સ્લીપિંગ મોડમાં છે?
ના સર, કમેન્ટ્સ તો આવે છે નો ક્વેશ્ચન, નો ક્વેશ્ચન, બટ નો
Yeah,
વન આસ્કિંગ એની ક્વેશ્ચન સર.
in general question answersમાં કેમ શરમાવ છો તમે? You can ask anything. Please feel free to ask the questions. The more and more questions you ask, the more and more clarity you have in your research. હવે આપણી એક્ઝામિનેશન બેઝડ વસ્તુ નથી કેરિયર. આપણું કેરિયર છે એ હવે સાયન્ટિફિક પાર્ટ તરફ જઈ રહ્યું છે, where when you try to have a clear mind, you can have a very clear vision what you want to do as a research in your PhD. How many students are live?
સર 15 સ્ટુડન્ટ્સ આર લાઈવ નાઉ.
Okay. So where are the remaining students?
સર
So normally they have the compulsion of to attend, or they can choose whether they want to attend or not.
यस सर, कंपलसरी फॉर अटेंड, बट क्या होता है सर, ये वीडियो भी अवेलेबल होगा whenever they get
Okay,
free or some have any issue, then they can watch that video in any time.
right. So apart from the normal live session, they can also go through this particular presentation whenever they find some time, or whenever they want to get more information out of this particular talk, right?
Yes sir, yes sir.
Okay, okay.
Okay sir, so we can end
So
the
if
session
there
now
are no
sir.
more questions, then shall we stop, or?
Yes sir, we can stop. Thank you so much for this session sir.
Thanks for the invitation, and I will be always happy to answer all the queries. So whenever in future you think that there is something which is required from my side, please feel
Sir,
free
one
to.
student is asking that, sir, could you please share the PPT to us?
Yeah, so as it is mentioned that this recording is going to be live and it is going to be available on YouTube, you can go through the YouTube channel. But if not, then I will be also happy to share this presentation with the students. Not a problem. By end of the week, I will share to the coordinator and you can ask the coordinator to get the presentation.
Yes,
Not a
sir. Yes,
problem.
sir. Okay, okay. Thank you, sir.
Thank you very much.
Okay, sir.